All right, let's go ahead and pick up where we left off. So we last left off, we were talking about channels, and there was an insert slide that I wanted to do for you guys. That's what I want to start off with. By the way, those of you who are online, if I sound muffled, it's because I'm wearing a mask. You can obviously hear it better. So I'm just trying to stay away from people. But if you have any trouble hearing, just let me know. Okay, so just to kind of review where we've been. All right, so we were talking about diffusion, and we're still in the passive transport section. We talked a little bit about simple diffusion. Now we're talking about facilitated diffusion. We talked a little bit about carrier proteins and how carrier proteins basically hand deliver molecules one at a time or two at a time, depends on how many they grab. But they're subject to saturation because they can only grab so many at a time. And if they fill up all of their pockets, then they can't do much more, right? So they're kind of limited that way. Most of what you see actually in passive transport is discussion around channels, right? And that's because they're higher output. You can kind of think of it, oftentimes the example I use is trying to move like cars across like a body of water, right? So like in the East, this is in the West thing. I mean, this is in California, so we don't have these sorts of things, but I know they exist in the East. Is every now and then you'll see like car ferries, right? Basically boats whose job it is to basically pack cars on there. They kind of sort of shuttle those cars across some body of water to the other side. And that's kind of what carriers are like, right? So they basically take a certain number of molecules, ferry them across the membrane. But the problem with that is the reason why it's lower output is because you're only limited to how many cars you can actually carry on the ferry, right? So if you can only carry 6 cars at a time and you're trying to move the entire population of, say, I don't know, the size of Detroit or something like that, then obviously it's going to take a while, isn't it? Just moving a couple of cars at a time. But the problem is you can't go any faster, right? Because you're limited in what you can do, and you're limited in how many you can grab. So that's what we mean by saturation limited, right? So once it's full, it's full. You can't go any faster than your max speed, and carriers are not fast. That's one of the reasons why we tend not to talk about carriers a lot in facilitated diffusion, because oftentimes in facilitated diffusion, what we want is high output. We want to get a lot of movement going on. And in a channel is what we usually typically refer to there. And that's kind of like building a bridge. Right, so if you build a bridge and it's big enough, then you can basically just kind of cram all those cars onto the bridge, and it's fairly fluid. They make their way across the bridge and you're good. So you can move a lot of people at once, certainly a lot faster than a ferry. And so that's kind of what you're usually looking at. And that's the reason why you tend to talk mostly about channel proteins in facilitated diffusion. But here's the problem. When you take a look at a channel is just basically like a pore in the membrane that allows usually some sort of a small polar molecule. That's usually what facilitated diffusion is for. It's for the movement of small nonpolar molecules. Excuse me. Rephrase that. It's for the movement of small polar molecules. All right, nonpolar doesn't need any help. They can already go across the membrane because the membrane's already nonpolar. So they don't have a problem with it. The ones that have a problem with it are the polar molecules and the charged molecules. So they're the ones who need a little bit of help. And that's what these channels are for. Now, each channel will be specific for its own molecule. For instance, a sodium channel is only specific to sodium. Calcium channel only specific to calcium. And so the specificity of it gives you this selective permeability. All right, so you can kind of pick and choose who you want to cross the membrane. So if you want a bunch of sodium ions to move across the membrane, then you pack it full of sodium channels. If you want a bunch of calcium, then you pack it full of calcium channels. Okay, so that gives you a sense of selection. And so the question then is, if you're talking about channels, remember you're talking about movement always from a high to a low concentration. If you recall, one of our last discussions was the energetics of moving from high to low, right? So moving from high to low is a spontaneous process because the universe is wired to always move from a high energy state to a low energy state. And so that going from high to low is always kind of an energy free sort of a system because the universe naturally wants you to go to your lowest energy state. But here's the thing, if you've got a concentration gradient, you don't necessarily just want any wild-eyed yahoo going through your channel, right? You want to have some control over that. Think about it this way. If you think about the holes in your wall at home, you don't just let anybody just come through your door, right? Your doors are regulated, they're controlled. So you get to pick and choose and decide who gets to come into your house and who doesn't get to come into your house, right? So there's a sense of selective permeability associated with your house. Now, here's the problem with that. All the channel is a hole. That's it. Just like all your doorway is a hole in your wall. Just the same thing as a window hole, same thing as anything else, right? So how do you control and regulate what goes through that hole? Well, that's the story of the slides that I want to actually go through with you when we're talking about the control of movement through a channel. Something I call channelology. So there's four main ways you can control movement through a channel. The first and the simplest is you control it with osmotic control. Now, in this particular sense, movement is controlled by a concentration gradient. So here's a membrane. There's a channel. It's going to be the outside and the inside of the cell. On the outside, you're going to have a high sodium concentration. On the inside, you're going to have a low sodium concentration. So if this first one is a sodium channel, which direction would sodium naturally move through that channel? Because remember, all diffusion, facilitated included, all moves from an area of high concentration to an area of low concentration. Right, so the direction that's going to naturally move is into the cell, right? Now, what about water? Which direction would water move? Well, let's take a look at water. Right, so water is moving not by diffusion, but by osmosis. And we know the mantra for osmosis, right? The mantra for osmosis is, yeah, salt sucks, right? So where's the salty side? It would be on the, well, we take a look at these two, which direction, which area is the salty area or the higher salt? Could be this one, right? Because it's the higher sodium, that sodium comes from sodium chloride. So which direction would water go? Yeah. Notice what you didn't do anything, did you? All you did to get these things to move through these little channels is all you did was manipulate their little osmotic environments. And this is typically how we use, this is how we get water to move through our body, through a channel called aquaporin. It's a special water channel. So how do we get water to move in versus out? It's very simple. All we do is we move the salt around. And we said we already know that salt sucks. Wherever we move that salt pile, it's going to suck water toward it. All we need to do is let water have free access. That's just go right through those channels in response to whatever that osmotic control is. Okay. So that's how we use water. The next three are three different types of what's called gated systems. An aquaporin, yes. So the first of the gated systems, there's three of them, is what's called a ligand gated system. So the ligand gated system is where a chemical signal opens the gate. So let's take a look at it here. Here's my membrane, there's my channel. And now this channel is gonna be gated. So there's gonna be like a protein gate on top of it. But in this gate is gonna be a little three-dimensional pocket that's gonna be specifically designed for your little chemical signal to bind to. So for instance, and that's a chemical signal, is what's referred to as a ligand. So you see these like little molecules that will then come in, and when they bind to the gate, it changes the shape of the gate. That gate will open up. And then if we have the same system set up, high on the outside, low on the inside, which direction is sodium going to run? Inside the cell, right? But only when you open up the gate. So it's going to run into the cell when you open up that gate. The nice thing about this kind of a system is the chemical signal you can create whenever you want. So you can have control over when this gate opens up or when it doesn't open up. Think of it as sort of like a lock on your door, right? So your door is like a gated system and the ligand is like the key. So you can determine when to unlock that access versus not, okay? Now you're probably thinking, where does the ligand come from? Well, if you think about it, remember when we talked about the secretory pathway? Well, some of those secreted proteins could be a ligand. So when you release that to the outside of the cell and it goes and talks to other cells, one of the ways it can talk to another cell is through something like this. It can be a ligand or a chemical like that actually communicates with another cell. And this is one way that you can communicate. Not the only way, but one way that you could. So the next one is what's called a voltage gated system. This is where a change in membrane potential opens the gate. Now let's take a look at this. So what I want to do is I want to do a ligand gate first. And then we'll hook it up to a voltage gate. Maybe a couple. So first of all, and it's the same system. So I want to keep the same system. So you've got high sodium on the outside, low sodium on the inside. So here's the thing. What is membrane potential? So basically a potential is essentially voltage. It's the voltage of the membrane. Due to positive ion distribution. So think about it. If you've got a concentration gradient of sodium, you don't just have sodium. You also have positive charge, right? Because sodium ion is a positively charged ion. So that means for every sodium that you have outside, you've also got a positive charge. What that basically means is that on the outside of this membrane, it's going to look slightly positively charged. On the inside, relatively speaking, it's going to look slightly negative. That disparity of charge separated by an insulator, in this case, which is the membrane, creates voltage or a membrane potential. We can actually measure this in muscle physiology. It actually comes in for skeletal muscles around about minus 70 millivolts. Now here's what we're going to do. First thing we're going to do is we're going to open up the first gate with a ligand. So we're going to bring in some sort of a chemical signal. It's going to bind to our ligand gate. And then what's going to happen? The gate's going to open, right? So once your gate opens, then what's going to happen? Well, as soon as as soon as this gate opens, what's going to happen immediately as soon as that gate opens? So the ligand's going to stay there because that's the only ligand gated. The rest of these are voltage gated. Sodium. What about the sodium? Yeah, right. Because it's like sitting there. It's like water behind a dam, right? As soon as you poke a hole in the dam, what's going to happen? The water's going to come blasting out, right? So, but this time, I don't want you to think of it in terms of sodium. Think of it in terms of charge, positive charge. So not only does sodium come rushing through, but what comes rushing through is all this positive charge. Well, guess what's going to happen? All that positive charge that was on the outside is now going to flip to the inside. And so instead of the inside looking negative-ish, guess what? It's going to start to look positive-ish, right? Because all these positive charges will basically go over and start making the inside look more positive than negative because all your sodium just came in. The outside is going to start looking more negative. So what you just did is you just flipped the charge around that membrane. That's a phenomenon in physiology called depolarization. You'll learn about that in AP1. So that's depolarization. That's essentially a change in the membrane potential. That's exactly what this gate, the voltage gate, senses. So when you flip the charge because of the inrush of positively charged sodium, that creates a membrane potential change that this gate now senses. And then what is it going to do? It's going to pop open. And then what are you going to see? Another inrush of sodium, or in this case, positive charge. And then that's going to make the piece next to it do the same thing. It's going to flip the charge. And then that's going to trigger this voltage gate to open up, and you're going to get another inrush of positive charge, which is going to do the same thing to the piece of membrane next to it, flipping the charge. And then on you go for as many of these voltage gates as you have. So notice what's happening. You kind of have this sort of almost domino-like sequential opening of voltage gates as this charge flip phenomenon rolls in that direction. This is because you have these voltage gated sodium channels that are sensitive to this voltage change. This, by the way, is the basic mechanism for how a neuron sends a nervous impulse through the body. So when you think of a neuron sending an electrical signal, the electrical signal is coming from the movement of these positively charged sodiums. It's basically the same set of physics as rubbing a balloon on your head. That positive and negative charge separation, that disparity. So just when you get to AMP1, act surprised. Okay. Or not. You can just get up there and say, listen, teach, sit down, I got this. Right? Let me take it over. Or not. Right? Don't do that. That most instructors probably would think that's cool, even though it would be kind of cool. But anyway. Okay, what's the last one? Mechanical gated. This is where some sort of a physical distortion opens the gate. So I'm going to go ahead and draw the channel. So here's my gate. Then on the gate is going to be a protein, looks like a little coil. And it's going to be attached to a distortable protein that's nearby. So if I distort this protein and I bend it this direction like that, then it's going to pull on this little coil and that's going to pop open the gate. And of course, in rushes your sodium. That mechanical gating system is how you hear. So your inner ear is actually attached. The sodium channels are actually attached to a distortable membrane. And then what happens is when that membrane distorts, it pops open the gates of those sodium channels, and then the inrush of sodium allows you to hear a particular pitch. If you open it up a little bit, that's one pitch. If you open up the gate a lot and get a huge inrush, then that's a different pitch. So different amounts of inrush of sodium is equivalent to or related to the different pitches that you hear. Now, how do you get the distortion? It's very simple. In your inner ear is fluid, endolymph actually. And so, and not only that, but the sound that you hear is literally nothing more than just compressed waves of air. That's all it is. So the compression waves of air from a sound that's being made will have different wavelengths to them and different energies to them. But when they hit your eardrum will vibrate as it kind of hits your eardrum. And then on your eardrum is going to be your auditory ossicles, which basically pulls on a membrane that's attached to the fluid in your inner ear on the other side. It's almost like a wave machine where you basically pull on that and you kind of create these waves that are equal in proportion to the air waves that are received by your ear from the sound. So when those waves wash over this membrane, it distorts it and it pops open the sodium channel. That's basically how you're able to hear pitch. So basically that's how you control movement through a channel, right? Through typically one of these four mechanisms. Either osmotic control, lagging gating, voltage gating, or mechanical gating. That's mostly what you see. So this basically is a channel. So you can see here you have your high concentration up here, low concentration down here. And then essentially you have your molecules coming in this direction. Alternatively, if you flip it around, you have high down here and low out here, then you can move that amount of flow in the opposite direction. So what this basically means is you have directionality. To a concentration gradient. So you can point them, right? Depending on how you build your concentration gradient, you can actually point these concentration gradients in different directions. So we've already seen how you can get diffusion rate differences, right, by the pitch or the steepness of the concentration gradient. Now you can actually get directionality. So it's actually quite helpful. And there's a cell that helps you to be able to get things in versus get things out. Now your carrier proteins, typically when you're moving things around, this is largely just for sort of background maintenance or things. So you're not generally looking at a lot of these. Why? Because they're limited by saturation. So that's basically if all of your transporters are full, then you're not going to get more, or you're not going to get more movement. And that's not necessarily helpful. Especially if you need like in a nervous response, like if you need that inrush of sodium in order to get your nervous impulse going, then doing things by carrier would not work, right? Because you wouldn't have a big enough response with the carrier in order to get that sort of massive depolarization that you need in order to kind of spark that kind of rolling effect that you see in a neuron. So you tend not to really look at carrier proteins unless you're working with active transport. Active transporters make most of their use of carrier proteins. Okay, so this is basically what a carrier protein looks like. So here you can see you've got basically one pocket per protein. So you're just basically grabbing it, you're shuttling it to the other side, you're letting it go, and you grab another one. So as soon as that one is full, then you're kind of out, right? So you don't really have a lot of capacity, you high output. Okay, the last bit of the passive transport is the osmosis piece, right? So with osmosis, remember, it all centers around two different things, right? Our solution, where water is our solvent, that's our solvent for life. We learned about that in chapter 3. And within the water, we have a lot of dissolved solutes, and the solutes are moving around by diffusion. And the solvent is basically moving by osmosis. And so ultimately osmosis is the diffusion of water across the membrane, chasing salt, right? Because salt sucks. And so salt is pulling water toward it. And that's basically always true. So here's an example of an osmotic experiment. So this is a U-tube. And so down the middle of the U-tube, we have a semi-permeable membrane that basically allows urea to pass. Excuse me. Allows water to pass. But not urea. So this is our urea molecule here. So you can see here, 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 and here, and there's only one over here, right? So this is going to, so which direction is water going to move? So if you have your high urea concentration over here, your low urea concentration over here, the direction of water is going to be moving where? So if we use the mantra salt sucks, or solute in this case sucks, then it's going to move in that direction. And you can actually tell that, right? Because you can see the difference in the volume. So here's one level of volume, here's the other level of volume. So you can see you've got a change in volume. So in order to equilibrate everything and make all the concentrations the same, water had to move to one side, and it had to add more water to the system in order to dilute it all. Right, so this is a system in equilibrium. These are equal concentrations as it currently stands, because this is after water is moved osmotically from right to left. Okay. But notice, if you use the mantra salt sucks or solute sucks, you will always be able to figure out what water is doing. Right, you just look for the concentrated area, and that's exactly where water is going to go. Okay, so since solutes have a sucking effect on water, then what that gives us in nature are three different salt environments. These are what's referred to as tonic environments. So tonic refers to salt. And so we have three different kinds, what we refer to as tonic environments. We have hypertonic, hypotonic, and isotonic. So if we take a look at a cell in its surroundings, in a hypertonic environment, the concentration of solute outside the cell is greater than the concentration of solute inside the cell. So which direction is water going to move here? If salt sucks, then water is going to move outside the cell and into the environment. In a hypotonic scenario, the solute outside is less than the solute inside. So in this case, which direction does the water move? Yep, it's going to go this direction, right? Chasing the salt, because now you didn't change the cell, all you do is change the environment. Right? So relative. It's relative to what's inside. But now in fresh water, the cell looks really salty. So that's where the water is going to go. And then in isotonic, your solute outside is equal to your solute inside. So water is going to be equal in this particular scenario. So there's no net exchange, there's no net increase. So water, we said, is moving in response to these three tonic environments, and it's moving through their own little channels, aquaporins, that are osmotically controlled. So one thing that oftentimes will throw students off, and this is kind of like the water balance slide, but when you take a look at cells, a couple of things you need to take a look at is what's happening with their water balance, right? So for instance, in this particular situation, cells have to deal with this. Why? Because if you have water inside of you, it's creating a hydrostatic pressure, right? This is the pressure of water. So there's a certain amount of water it's exerted, or pressure it's exerted by water. That's hydrostatic pressure. And the problem is, if that pressure is too strong and you don't have mechanisms in your biology to deal with that pressure, that could be catastrophic for you, right? So for you, being able to deal with the strength of the water pressure associated with osmosis is really critical, because if you don't have mechanisms to deal with that, it could kill you, right? And so when we take a look then at this water pressure idea, what we're really talking about is what's called osmotic pressure. Osmotic pressure is defined very cleverly as the force needed to stop osmotic flow, whatever that is, right? So this is basically something that we do to sort of measure the force of osmosis. And it's one of those things where you can't really measure it directly, right? So when water's coming at you, you can't really measure it directly. The best thing you can do is oppose it and then measure what you're using to oppose it, right? You're using one of those Newtonian physics laws of motion, right? For every action, there's an equal and opposite reaction. Remember that one? That's like high school physics, right? Well, what that basically means is for forces that we can't measure directly, we can measure them indirectly by using that principle to oppose that force. Here's a good example. Let's say I wanted to measure the weight of the door, the force that the door has as it's closing. But there's a certain amount of force that it's exerting, but I don't have a way to measure it directly. Well, one thing I can do is I can oppose this force coming at me with my own force, right, with my own hand. And the point at which it stops is the point at which the force I'm pushing on the door is equal to the door pushing on me, right? That's using the equal and opposite reaction. So if I can measure my force that I'm pushing against the door, it's going to be equal to the force that the door is pushing on me. So when you're measuring osmosis, right, the pressure, osmotic pressure, you don't just measure the pressure like a tire gauge. You say, okay, there it is. What you do is you oppose the flow of water. And the amount of force you have to exert in order to oppose the water is equal to the amount of pressure that the water is pushing back on you, right? That force is what's called osmotic pressure. Now, the reason why it's important is because the idea of osmotic pressure is actually directly proportional to the salt. So osmotic flow is pulled by the suckiness of the salt that it's responding to. So like, for instance, if there's like a salt pile right here that's pulling water toward it, and I wanted to know how strong is your sucking effect on water, then what I would need to do is stand in between there as the water's coming at me and oppose the flow of the water. The force that I use against the water is equal to and directly proportional to the strength of that sucking effect of the salt. So the more suck that the water has on it, the stronger that osmotic pressure is going to be. If it's a very gentle suck, then it's not going to be a very strong osmotic pressure. So the osmotic pressure, remember, exists because of the suckiness of salt. It's directly going towards salt. And so it is related to the concentration of salt. Okay. So what happens if you have some of this water flow stuff going on and you're an animal or you're a plant cell? Well, depending on who you are, your fate's going to be different, right? So let's take a look, first of all, at the hypertonic solution. Remember, in the hypertonic solution, the salty area is on the outside of the cell. So that means all the water is going to leave the cell, dehydrating the cell behind it, and it's going to get all shriveled up. For an animal cell, that is not a good situation. That's dehydration. By the way, if it's a plant cell, it's not any better, right? Your water is leaving your vacuole. And not only that, but your membrane is sucking here in the middle. So all this stuff is bunching up in the middle, and you got a lot of slack in these cell walls. So this is like a flimsy little piece of cardboard now. So this is a plasmolized cell. And this is basically a plant that is pretty much on the ground. I mean, it is in bad shape. So a hypertonic environment for a plant isn't any better than it is for an animal. Well, let's see if we can improve this situation. Let's go to isotonic, shall we? For an animal, isotonic, you have equality inside and outside. And so you have equal exchange of water. And everything is nice, right? So you have neither gain nor loss of water. Everything is well hydrated. The cells are nice and poofy and plump. And as an animal, you're quite happy. This is a good place to be isotonic. What about if you're a plant? Well, if you're a plant, I mean, are you better than plasmolized? Sure. But you still have a lot of slack in these walls. There's really no pressure against these walls at all. So this is what's referred to as a flaccid cell. It's wilty. So this would be a plant that's kind of like that. Is that a happy plant? No. Well, let's go to hypotonic. And hypotonic, remember, your salty environment looks salty on the inside. So it's going to suck all that water into the cell and it's going to blow it up like a water balloon and it's going to burst it. So this basically creates an anemia state, a loss of red blood cells. That's obviously a bad thing. If you're blowing your cells up, that's not a good situation. Okay, so this is very much not okay for an animal. But what if you're a plant? Well, if you're a plant, you have all this water coming in. It's really expanding your vacuole. Your vacuole is really pushing against the cell wall. So you can see how the cell walls are kind of bowing outward a little bit. So they're kind of bowing like this, a little outward. So this becomes very hard, rigid cell walls. It's what's referred to as a turgid plant. So this plant is really nice and perky. Very happy plant. So this is where you want to be right there. You want to be in a turgid state for a plant. So notice the consequences of each one of these is a little bit different depending on whether you're a plant or whether you're an animal. So if you're an animal, you need to be isotonic. If you're a plant, you can actually deal with a little bit of flexibility. Now, is this plant over here dead? No. You can actually change this. So, matter of fact, if you flush this out with fresh water right after it, then it'll just return right back and it'll be fine. If it persists in this state, however, yeah, it's going to be it's going to be a situation. But you're going to dehydrate the plant and the plant's going to die. So you can see how you have different types of outcomes depending on whether or not you have that nice cell wall. And that's the reason why osmotic balance is one of the big biological questions of life. You need to be able to balance yourself osmotically. If you are a protist, remember we talked about contractile vacuoles, basically bailing water out of your cell so that you don't blow up like a water balloon. So that was important for paramecium to survive, right? So that was a huge question that I had to answer, that water bound, the osmotic balance situation. If you're a marine organism, you have to answer it by basically making your internal concentration match the seawater. So you're basically calibrating your own internal saltiness to match the saltiness of the ocean that's around you. Or if you're a freshwater fish, to match the relatively non-saltiness of the freshwater that's around you. And if you're a terrestrial animal, like us, for instance, you don't have to manage, you don't have to calibrate your environment, internal environment to external. What you do is you circulate isotonic fluid. What's our isotonic fluid? Our blood throughout our body. So we basically bathe our tissues in isotonic fluid. That's our body fluids. Right? So all of your body fluids are isotonic in nature. And so that's what you basically bathe your bodies for. So you have like you basically have kind of like it's kind of a cordoned off system of isotonic solution. So it's separated from the outside world so that you don't have to worry about what your outside world looks like. You basically have an internal system that's blocked off and separated out from your external system, external environment. So that basically ensures that you have kind of a self-regulating internal environment that you can control regardless of where you are, which is one of the reasons why we can go swimming in the ocean where it's very saline, or in a freshwater river where it's not very saline at all, right? If you take a saltwater fish and you put it in fresh water, it's going to die, right? Why? Because it has to calibrate its internal fluids to look like it's external. And if it's normally salty on the inside, all that fresh water is going to flood into it and it's going to blow up its cells. And then that's why it starts floating after a while. Right? So, by the way, the push or the water pressure against the cell walls that we see is what's referred to as turgor pressure. This is basically the water pressure. Pushing against the cell walls. Okay. Any questions on passive before we move into active? So there's two types of active. Remember we said there's ATP driven, and then there is concentration gradient driven. And so we're going to take a look at ATP first. So this is basically using ATP to directly fuel active transport. That's the one we're going to be looking at. In this case, in active transport, because it's an uphill process, it's going from a low to a high. So it's going to a less energetically favorable state. And remember, we said that whenever you have a channel with a concentration gradient, always remember that what got you that concentration gradient was some sort of an active transporter somewhere, right? So if you have a sodium concentration gradient, it was a sodium transporter that got that there in the first place. They come together. So you don't just get these concentration gradients for free. You have to build them. And this is the way that you build them. So these are all carrier proteins. Active transporters typically are. And so this is where we talk about our carriers mostly. And you have different types of carriers. So a uniporter is basically a protein or a carrier that will basically take a molecule and move it in one direction. So one molecule at a time in one direction. A symporter is when you're moving two molecules, but they're going in the same direction. So if you take a look at a transporter, you'll have one molecule going this direction, another molecule going the same direction. That's a symporter. So you're moving them in the same direction at the same time. An antiporter is where you're using two molecules, but it's going in opposite directions. So if you take a look at an antiporter, in this particular case, we're going to use circular molecule coming in, triangular molecule going out. So that would be an antiporter. So it's two-way traffic. And so that's kind of what you usually have. And you can have a combination of the two. Like if you get into AMP2, physiology half, you see symporters and uniporters and antiporters. You see all different versions of these different active transporters. They're kind of all over the place. But the poster boy that we're going to take a look at is an antiporter that is an ATP-driven pump that is responsible for moving sodium and potassium. So it'll take 3 sodiums and move it out for 2 potassium to move it in. Both of them are going against the concentration gradient, right? So here you have 2 concentration gradients that are kind of pointed against each other, right? So the high side of potassium is on the inside, the high side of sodium is on the outside. So they're pointing in the opposite directions when we're talking about directionality, but they're both going to be moved with the help of ATP, right? So that's your energy molecule. So let's take a look at the mechanism that we have here. There's a couple of steps. We just want to make sure we unpack it so we can see what's happening. The first thing to understand is starting off at number 1. This is what we refer to as our native conformation. So it's our native shape. And in our native shape, you have a particular set of systems. So there's a couple of pockets here that we see. So our sodium pockets are nice, cushy little chairs, very comfortable little things in this particular shape. And this will allow sodium to bind to these nice, cushy little sites. And that's exactly what happens. So sodium comes in here, binds to each of its cushy little chairs. But in the native conformation, the potassium seats are not available. Matter of fact, they're quite inhospitable. Kind of like, you know, it's like when you go to take a look at the potassium chair, there's like a bunch of knives sticking out of it, right? You're probably not going to want to sit in that one, not unless you like to have your backside, right, diced up. Which you probably don't. So we're just going to assume you don't. If you do, don't tell me that's a tale for a different class. But basically, it's inhospitable, right? And so the first thing that happens in this mechanism is the sodium will bind to its inviting, nice, comfy little chairs. And then you basically hydrolyze some ATP, and you're going to get a little bit of an energy kick. Now, what happens here is you're going to get a shape change because of the energy that's built into this phosphate. So ATP will hydrolyze, ADP will leave, it'll leave the phosphate behind. That's packing enough energy to cause the shape change. What does it shape change into? It shape changes into this guy over here. This is the alternate conformation. Now let's take a look at the alternate conformation. And the alternate conformation is just the opposite of what the native conformation looks like. And now all of a sudden, the sodium chairs are not very hospitable, and the potassium chairs are. So in its alternate conformation, what's happened is the potassium chairs have become nice and inviting, soft and cushy. Right? Just think about it, long day on your feet. You see one of those like really nice kind of melty little chairs. Maybe if you're one of those massage chair type people at the mall, spending all day long walking around, and you see one of those chairs, and you can't help it, right? You're like, how can I sit in that? Because I got to get off my feet, right? At this point, I got to get off my seat so bad, I'm willing to just sit on the ground. Right? But you're going to sit on the chair first of all, and that's kind of what's happening here. So now all of a sudden, you got this nice cushy little chair for the potassiums. But guess what's happened to the sodium during the shape change? As it's been sitting in its nice little chair, all of a sudden, when the shape started changing, knives started coming out of the seat. And if knives are coming up out of the seat into your butt, how long is it going to take for you to stand up? Yeah, you're going to be up quick, right? And that's exactly what happens. So these guys get kicked out, the sodiums, and they get released to the outside. That's where the gradient is. Then after that, you got your potassiums that will come in and be like, hey, take a look at this. I've got some nice cushy little chairs. I'm going to come in here and sit down. And so it's going to sit down. It's going to be all nice and happy, but these are inaccessible. These are unavailable to the sodiums. So the next step is once your potassium binds, then you're going to release your phosphate. That's your energy that's maintaining your shape change. And you're going to go through another shape change back to your native state. But guess what your native state looks like? The knives come out in the potassium chairs now. In this particular case, now the potassiums be like, no, I'm out, right? And then the nice soft cushy chairs are back. Sodium. And then what do? In your native conformation, you invite sodiums to sit down in your nice cushy little chairs again. And you do it all over again. Right? So basically what you have is this kind of like a mechanistic cycle, if you will, of shape change. So on one side, you have your sodium potassium pump. It's going to be available for sodium. It's going to pick up the sodium. Then it's going to hydrolyze the ATP. It's going to go through a shape change. Once it changes shape, it basically dumps the sodiums out on the other side, grabs the potassium, and it goes through a shape change. Then it dumps the potassium, grabs the sodium, and goes through a shape change. Dumps the sodium, grabs the potassium, goes through a shape change, and so forth. Just completely like this is what it does. It's just like change the shape, grab a dump, grab a dump, grab a dump, grab a dump. That's basically what it's doing the entire time. So you can see that after a couple of rounds of that, you're going to start building up a nice little sodium concentration gradient. But what about the indirect version, the so-called coupled transport that utilizes a concentration gradient? Well, this one's a little bit different, right? Because it still uses ATP, because ATP will indirectly create this. We'll see this here in just a second. But directly what it does is, and it uses the glucose sodium symporter for this one, is it's going to use the concentration gradient of sodium to drive the symport of glucose. All right, so when you take a look at this, here's your sodium potassium pump over here. So you can see you're creating your sodium concentration gradient over here. And when your sodium starts to move inside, it releases a lot of energy. And that energy, as it releases, will power the glucose up its hill, right? So the energy that's being released by the sodium going down its gradient is transferred to glucose to power the glucose up its gradient. So let's think about it in terms of an anti-Borders, right? That's what I like to use. Let's imagine that we're two hikers on a hill, and I'm going to pick on Kelsey since she's closest. Right, and so let's say Kelsey's going uphill. She's got like a 100-pound pack on her back, and I've got one on mine, but I'm going downhill. So I'm done with my trip. I'm getting ready to head home, and I'm coming down the hill. Well, we've probably been on enough hikes to realize that the upward trip is a whole lot harder than the downward trip, right? Because the downward trip has a lot of release of energy associated with it, a lot of momentum associated with it, right? So as I'm going down a hill, I've got a big pack on my back anyway. I mean, really, literally what it is just me sticking my foot out. It's like a controlled fall, right? I mean, it's like I'm just falling down the hill. All I really have to do is just stick my foot out and just make sure I land on my foot, right? That's about it. So you're more like just falling. But as you're going down the slope, you're releasing a lot of momentum, aren't you? You can actually feel it. Sometimes if you've got a heavy pack on your back, you can kind of feel yourself getting a little out of control, right? You're going to have to sort of put the brakes on and kind of push back with your legs a little bit because you can actually feel that energy that's trying to come out of you. Well, here's what's going to happen. I'm going to be coming down the hill with my big pack on. And I got all this momentum coming out of me, all this energy coming out of me. And that's what's happening with this concentration gradient, right? We're going downhill, they're going downhill. And so they're releasing all this energy. But now what I want to do is I want to basically use some of my energy because I got it for free, right? I mean, it's just there for the taking, right? I mean, I don't have to do anything for this momentum, it's there. But Kelsey could do some, right? Because she's having to fight her way up the hill with this 100-pound pack. And it's slow going, right? I mean, the steps are smaller, it's a bit more trudgy. So as I pass her, I'm gonna do kind of like one of those little martial arts moves, right? Where you sort of do a body, a momentum transfer, the other person's body to get them off balance. And I'm gonna do that to Kelsey. I'm not gonna put her on the ground, but I'm not even gonna touch it, make it closer. But basically, as I pass by, imagine that what I do is I transfer some of my momentum to her, like judo style, right? And then as I transfer that momentum to her, I kind of push her uphill a little bit. I sort of use my energy and I channel it into her, and I kind of use that to kind of slingshot her uphill a little bit. Well, for those next couple of steps, they're a little bit easier for her, right? So there's not quite as much energy needed for her because I transferred my energy into her and kind of gave her a bit of a boost, a bit of a shove, right? So those next couple of steps are a little easier, energetically speaking. That's exactly the kind of transfer that's happening here. So when you have a concentration gradient where one molecule is going down, it's like it transfers that energy to the other molecule that's going up. And basically it fuels and powers that molecule's ability to get up the hill. Okay. The last of the groupings, the bulk transport. So we have two major types of bulk transport. And the bulk transport, the reason why a lot of times students will be like, wait, I don't understand the difference between, say, for instance, bulk transport and some of the others. Why isn't bulk transport considered an active transport or something like that? What sets bulk and the other transports apart from each other is the bulk transport is the movement of large macromolecules. So notice what's happened up to this particular point. Every single time I've made sure to mention that we're moving what? Small polar or small non-polar molecules, right? That's basically what we've always said. They've always emphasized smallness Now we're talking about large macromolecules. So you can't just put a channel in your cell and move a big molecule, because what's going to happen is it's going to just blow too big of a hole in the cell membrane. It's going to compromise its integrity. So you have a different system. You have endocytosis, which is movement into the cell. You have exocytosis, which is movement out of the cell. And so when you take a look at the different types of endocytosis, so this is phagocytosis, we talked about this already, where you can engulf, like in this case, a bacterial cell, like this, I think, is a white blood cell, it's engulfing a bacterial cell. So you can see that you basically kind of start to suck it inward, you start to kind of pucker your membrane. And as you kind of continue to pucker your membrane, what's going to happen is if these shoulders touch, then you're going to pop it in. And you're going to basically surround it with a membrane, and then it's going to be what you've got. We can use your lysosomes to that, and you can just basically devour that bacterial cell. And that's kind of what's happening here. So you can see this guy's almost not quite there just yet. Right. So it's got a little bit, almost there, about halfway there. So a little bit more, then you'll have that thing engulfed. So that's phagocytosis. That's one means of endocytosis, and then penocytosis, which is cell drinking. Where cell drinking basically is when you're taking small little sips. So you're basically just doing the same thing as you do with phagocytosis. The only difference is you're just taking whatever happens to be out here in your extracellular matrix. Right, so that's your extracellular matrix out here. So you can just kind of suck in whatever solutes you have out there and just sort of pinch it off, and you're going to get whatever a sampling, if you will, of whatever solutes you have out there. So you see like a lot of these little peanut, they're smaller, but they tend to have a lot of them. So there'll be like a nice little row of them. And this is helpful for the cell. If the cell has to change what's in its extracellular matrix, if the cell has to sort of modify some things out there, it's kind of helpful to know what's out there in the first place. Like how much proteoglycan do I have out there, right? How much ground substance do I have out there, that sort of thing. So the good thing you do is do that, a little bit of that cell drinking sort of a thing, just to kind of sample some of that stuff that's out there, and you can kind of get an idea of what you need to put out there if you don't already have it out there. And then your receptor-mediated endocytosis is a similar sort of a situation. Only in this case, what you do is you put receptors out there that are specific for your target molecule. So this is like when you want to cherry pick out specific things from your external environment. And so in this particular case, you'll grab onto these specific molecules. But in order to pull these in and internalize them, you don't want to just pull on a receptor like that, because that's just going to tear it through the membrane, right? So you're just going to tear it through the membrane. That's not going to be helpful. So what you do is you basically tether your receptors into a little protein foundation called clathrin. And this protein foundation kind of coats the inside of this and sort of embeds these receptors in there. So then all you have to do is just pull on this clathrin foundation, and it kind of pulls on the entire membrane. So you're literally just kind of like pulling this membrane and sort of causing it to pucker. It's a slightly different mechanism than the others. And then what you end up getting is this clathrin coated vesicle full of these very specific molecules that you selectively pulled out. And so here you can see this little blue haze. So here are some beginning clathrin coats. So you can see these little clathrin foundations here. That's what these little hazes are. And then on the inside, you can see these are the fully surrounded clathrin coats. So the blue is the coating associated with the clathrin itself. So it's a slightly different mechanism, helpful for when, say, for instance, as a cell, you want to just grab onto something very specific, very intentional. The exocytosis is basically the reverse of that. Right, we saw this one with the secretory vesicle, right? So when we saw the secretory vesicle fusing to the plasma membrane, that was exocytosis. So that basically fused and sort of dumped everything on. That's kind of like two fusing soap bubbles. So when you have one, your secretory vesicle fusing with the membrane, it's going to attach, and then it's going to basically dump all of its contents to the outside of the cell. And so this is actually a really perfect use of an exocytotic vesicle. So you can see it's actually spewing outward. It's basically emptying out Now, this is something we already saw. Okay. So for you table people, you like to study with tables and organize your information with tables. These are your tables. So these are your mechanisms for transport. You see facilitated diffusion, carrier, and channel driven. You see your active transport, your osmosis, and then you see your endocytosis, both phagocytosis, tenocytosis, receptor mediated, and exocytosis. That's chapter 5. So the next chapter is going to be chapter 10. Because 6, 7, and 8 are part of the take-home group. Okay. Chapter 10. So this is mitosis, right? So cell cycle. And when we take a look at this is like the entire time, the entire semester, we've been talking about how, like, you know, the ultimate goal for all biological organisms is reproduce, right? But it's like almost over, and we haven't even talked about reproduction. So if it's so important, then how come we never talk about it? Well, it's because you have to do a lot of work before you get to the reproduction piece of it. But this is the first chapter, actually, where you can actually talk about reproduction. So this is actually a reproductive cell. I mean, this is actually a reproductive chapter. Right? So if you are a single cell, for you, this is your reproduction chapter. Now, for us, we got a little bit more work to do because we're multicellular, so we have to do a few extra things. But if you're a single cell, this one here, this is your reproductive chapter. Okay, so we're talking, first of all, about cell division. And cell division is something that happens in all organisms, right? Because after all, in order to grow, we have to do cell division. We have to make more cells. In order to reproduce, we have to do cell division. Right? As we get older and we stop growing, we still are going to damage our tissues, right? So we have to do wound healing. That's going to require cell division. Cell replacement. Cells just naturally senesce. They don't live forever. So you're going to have to replace some of your cells as they get old and worn out. And so that's cell division. So cell division is something that's with us all the time. So for us, it's focused on these four right here. For a prokaryote, which is a single-celled organism, reproduction equals cell division. Right, so it's really, really important for all organisms, because without cell division, we just don't have the building materials that we need in order to build our body. Remember in the cell theory, we heard about in chapter 4, or we said that part of cell theory was that all living things are made of cells. So if you're made of cells and you're made of a lot of cells, like us, for instance, then it makes a lot of sense that you're going to have some sort of a strategy to make lots of cells. So there has to be a cell reproduction type of strategy associated with it. So let's take a look at bacterial cell division. We're going to take a look at the simplest first, then we're going to get more complicated. So with bacterial cells, they are single-celled organisms. They don't have a sexual life cycle, so they are basically clonal in nature, and they divide by a process called binary fission. We're going to take a look at this here in just a second. So in bacteria, their genome is a huge circle. So for instance, that genome that's in the nucleoid region is a giant circle. It's just kind of all folded on itself. And then the first thing you have to do in cell division is you have to basically go from one parent cell to two daughter cells. Now, in mitosis, these two daughter cells are identical because you just need to make more of the same. So let's kind of back it up a little bit and sort of sketch this out, because it would probably be helpful to sort of see how it all fits together. This chapter typically fits well with the next chapter and the next chapter after that. Matter of fact, 10, 11, and 12 are kind of a triumvirate that typically go together. Because genetics, which is chapter 12, is rested on a mechanism of meiosis. And that's chapter 11. Right. So in order to understand genetics well, you have to understand meiosis. Understand meiosis, you have to understand mitosis. And so that's the reason why these three go together. So let's take a look first of all at how this all works together. So generally speaking, we all start off as a single cell with 46 chromosomes. This is you. So the first thing we all do is we start going through multiple rounds of mitosis, which is this chapter. These are two genetically identical daughter cells. And then they will go through cell division. Who will then go through cell division. Will then go through cell division. And so forth. You get the idea. And then eventually we'll build a body that's big enough and then we're born. A little diaper. Right. So notice this is all mitosis. So mitosis, you're basically generating two identical daughter cells. Okay. So that's what this chapter is about. Cell division. For us, it's how we build our body. For an individual single-celled organism, it's how they reproduce in the first place. But as it turns out, it's all the same because they have to go through the same cell cycle that we do. The only difference is when they finish a cell cycle, they've successfully reproduced. When we finish the cell cycle, all we've done is added more cells. We got a little ways to go first before we successfully reproduce. Okay. Now, the way that this works is that a subset of these cells in the baby's body are set aside, and they go through a different type of cell cycle, which has two phases to it, called meiosis. And there's a meiosis 1, the meiosis 2. And what they do is they create 4 genetically non-identical daughters. They're all different. And then notice they're half the number of chromosomes. So we have 46 chromosomes, one from mom, one from dad, right? We learned about that when we did the mitosis chromosome squash, right? You have 2 chromosome ones, one from mom, one from dad, 2 twos, one from mom, one from dad, so forth. So you have 46 altogether, right? 22 autosomes times 2 is 44 plus an XX or an XY. That gets you up to 45 and 46. So during meiosis, what we do is we have that down to 23. So then when this 23, say from mom, meets 23 from dad, then these two will fuse together to give you what? 46. And a single cell with 46 in it. So there's the next generation that starts off life the same way we did, as a single cell with 46 chromosomes, which for the next 9 months is basically busy doing only mitosis, building your body. Okay. So if you're wondering, you just got the birds of the beanstalk. Not that you need it, but to say it. That's kind of the nerdy version of it, but that's definitely what that was. Okay. But now you can see, though, you can see how mitosis fits together with meiosis in us, which are a multicellular sexually reproduction organism, right? So meiosis is what produces our gametes, our eggs and our sperm. Mitosis is what builds our body. Okay, that's our body cells. And then the genetics piece of it comes into play when you talk about the generationality of it, right? So basically the eggs and the sperm of one generation is carrying the traits that will carry forward to the next generation. And then they will then do the same thing, which will then pass on the traits to the next generation, to the next generation, next generation. So when you're passing traits on from one generation to the next, that's genetics. That's inheritance. So you can see how the 3 chapters they really go together as kind of a package deal. So that's kind of where it brings us in, this idea of having one cell that creates these, in terms of just the mitosis piece of it, that creates these genetically identical daughter cells, right? And that's kind of what happens with bacteria. You get these 2 genetically identical, or clonal in this case, daughter cells. And once you've done that, then you successfully replicate it. But here's the thing. With cell division, there's a mantra about it. And it goes in this order. Duplicate, then separate. So basically what you're going to do is you're going to essentially take a cell and you're going to split it in half. But in order to do that, you've got to double everything first and then split it in half. Because what happens if you didn't double everything and split it in half? Well, if you didn't double it, then what you would get is 23 in the next generation. And then when these guys did the same thing, what would they give rise to? 12 and a half, right? And if these guys did the same thing, what would they give rise to? 6 and a quarter. So you can see how you're diminishing every generation. If all you did was have it, then you would be diminishing every generation. And within a few generations, you'd have no more DNA. So if you want to keep everything the same, like for us, if you want to have cells that are all the same, then you have to double everything and then have it. So that doubling then is the required first step before you separate it into two equivalent piles or two equal piles. So let's take a look here. First of all, the replication, which is chapter 14, we'll take a look at that. We'll take a look at that one. But basically what we notice is that on bacterial cells, these little circular chromosomes, we notice that there is a start site for replication, for duplication. We call it the origin. That's the green. It's like a flagged area on the chromosome that says, here, start here. When you go to duplicate, replicate your DNA, start here. Now, bacteria also have this red termination site that's kind of like a stop sign. And so what happens is the replication enzymes will roll in both directions, counterclockwise and clockwise, into what's called the double rolling circle, and then eventually they'll meet at the termination site to form what's called the linked magician's ring stage. I think that's what I call it anyway. Right, and then eventually what's going to happen is you're going to separate that termination site into two separate genomes. And then as soon as you elongate the cell, all you have to do is just pinch these guys apart. You've got two brand new cells. You successfully duplicated everything. They're carbon copies of each other. Okay, so what you just did now, where you had one cell, you now have two. And if these guys double, then where you had two cells, you now have four. Right, we have 4, you have 8, 16, 32, 64, and so forth. And you'll get a nice little colony of cells. And we're not going to get hugely into bacterial, replication genetics and stuff like that, but there's a lot more to that story, but we're not going to get into that. So what about the separation? Well, what happens once you actually create the replicates of these two genomes, then you basically get this kind of little pucker area that's connected by a ring of proteins called a septum, and then you just kind of pinch these guys apart into separate pieces. So you literally just kind of cinch it together, just kind of pinch it together, just like a drawstring, right? You just kind of cinch it together, and it basically pinches them apart. The protein that's responsible for that is a protein called FITC. And FITC kind of forms this little ring-like structure, and we're going to take a look at that here in the next slide. So here's a real bacterial cell where you can actually see the septum. So this is the actual septum here. And you can see this like little contractile ring. And this is made of the protein FITC. So you can see it's just this like little ring, like this little drawstring ring that's around that little central area. And then what happens is it's contractile, it'll cinch down, right? Just like a little aperture, it'll cinch down. And when it does that, then it pinches apart the cell and it pinches it into two different cells. And this is a protein that we find in most of the prokaryotes that we studied during cell division. So it's very simple. You replicate, right? That's your duplication piece of it. And then once you do that, then you separate. And it's separation is that little contractile ring of FITC that just basically pinches them apart. Well, FITC, the protein FITC is interesting because even though the prokaryotes have FITC, the eukaryotes don't. So the question is, what does it for eukaryotes? Well, we noticed was even if they don't have FITC does actually have a similarity to a protein that we do see in eukaryotes. FITC is very similar to tubulin. And we already saw what tubulin does, right? Tubulin builds as microtubules. And those microtubules will basically help you separate stuff and move stuff around in the cell. So FITC is perhaps, I mean, if you want to think of it like in terms of evolutionary terms, you could probably think of FITC as maybe a precursor to, is like an evolutionary precursor to tubulin, perhaps. But here you can see two different proteins, but similar function, which is one of the problems why oftentimes you kind of have, kind of a little bit of a muddling of evolution, especially when you take a look at things like morphology or function, right? Because sometimes what you can have is or what I just stated is just a hypothesis. It's like, well, maybe FITC is sort of like the evolutionary precursor to tubulin. So maybe FITC evolved into tubulin. But the problem is we see actually a lot of situations where that's never the case, where the FITC and tubulin are actually have, they don't structurally have anything to do with each other, but functionally they serve the same function. So it's almost like two completely different things that have nothing to do with each other, actually end up serving the same function in two different cells. And so that kind of muddies the water when you're looking at evolution from a morphological or functional sort of an evolution sort of a perspective, which always is those kind of nice little wrinkles that you see in evolution. So let's take a look then. So we already saw the binary fission with the FITC on the left-hand side, right? But let's take a look and see what the other organisms do when they're dividing themselves up, right? Because you have a couple of things here. Obviously, if you're a eukaryote, you've got a big problem, first of all, right? That nuclear envelope, which is designed to protect your DNA, that is a problem, right? So here's the thing. It's a problem not only because it's in the way, but it's a problem because it's there for a reason. Or the nuclear envelope is there to protect your DNA from bad guys. Who are the bad guys? Pretty much everything else around you. So if you are basically in an environment where there's danger everywhere, are you going to take your protection off? Probably not, right? You're going to try to figure out how to do what you need to do with your protection up and in full array, right? So if you take a look at these, right, we're talking about some protists here and yeast in particular. So here's a couple of protists, different types of protist patterns that we see. So when we take a look at separating the nucleus around in some protists, what they'll do is they'll have kind of different microtubule organizing centers associated with them, but they don't ever, they're not going to ever break down the nuclear envelope. They keep that nuclear envelope intact. What they do is they have kind of these thoroughfare microtubules that go through it, but then they have these other side microtubules that kind of grab onto the chromosomes, but notice they don't ever let go of that. There's a bit of a delay going on here. Let's figure out what's going on. That took long enough. Okay, that was like about a good 20 minutes there with the delay. In some of the protists, you'll notice, so you'll have what looks like centrosomes, right? These little centrioles. So these would be like the animal-like ones. So some protists are animal-like, like amoeba, for instance. Paramecium is animal-like. And some are plant-like, right? Like Euglena is plant-like. Some of the others are plant-like. So this would be like an animal-like one where you have a centriole. It does the same basic thing. Notice it kind of creates this like thoroughfare, and then it kind of has these offshoots that kind of pokes into the membrane in the nuclear envelope and it grabs onto the chromosome. So without that, then you're talking about the plant-like ones. And then there's yeast, right? So yeast, it doesn't break the membrane down. It actually builds what's referred to as its spindle body, right? Which is that array of microtubules that are coming off of the centriole. It does that inside the envelope. So you can see it kind of creates this like little protein pad here, which is the spindle pole body on either end. And then from this, it's going to build microtubules, some of which are going to grab onto the chromosome, some of which are going to grab onto each other and kind of push off from each other. But notice you never break down the nuclear envelope. So think about it. This makes total sense, doesn't it? What is your most precious molecule in the cell? The one you can't do without. Huh? Yeah, your DNA, right? Because your DNA has all the information that you need in order to do whatever you need. You've got that, you've got everything you need, right? You don't have that, you are screwed, right? You have nothing to run on. So if you're a protist, a single cell protist like a euglena in the middle of a pond, what is life like for you? Is it fairly cushy and comfortable and peaceful? No, it's a pretty dangerous world out there, isn't it? Same thing if you're a paramecium or you're an amoeba, dangerous world, yeast, dangerous world out there, right? So what you're looking at are really single cell. These are all single cells for the most part. And you can see that there's a reason why they don't tear their nuclear envelope down. Because to do so would be titanically risky, right? Because the second you tear down your nuclear envelope, you're losing all that protection of your DNA. And if you screw up your DNA, you are toast, right? So it's easier for you to protect your DNA and more important for you to protect your DNA amidst a pond full of enemies and things that want to kill you and eat you. It's more important for you to protect your DNA. You can figure out how to kind of rig separating that, the cell cycle, but the premium, the priority is protecting the DNA, which is the reason why they don't break down their nuclear envelope. Now let's take a look at us. Look at what we do. We take our nuclear envelope right on down, right? We just dissolve it all together. We build our spindle pole and we pull our chromosomes to either end. We don't care. How can we get away with that? And protists can't. Yeah, we're not single celled, right? So we're not a single celled organism fighting to protect its DNA in the middle of a dangerous pond. Our cells, which are going through mitosis, are usually protected and internal amidst other cells. So they're not around enemies, they're around friends. Okay. So we can afford to be a little more risky because we've got more protection. We've surrounded ourselves with friends. Right? It's like the safety in numbers idea, right? So you're not exposed there by yourself. So that basically brings us to this this idea of the eukaryotic chromosomes. We kind of already know this one, right? So we already know that we have 46 chromosomes. We have 23 pairs, 22 autosomes. That's 1 through 22. And then 2 sex chromosomes. That's XX or XY, right? That's what gives us our 46. But ultimately, if we have like missing chromosomes, these are actually real chromosomes. This is colorized, by the way. They don't come in blue and red. That's not, chromosomes aren't colored. So they're just, you know, whatever they are, black and white basically is what they would be. But this basically shows you like chromosomes, what chromosomes look like when they're actively dividing. Okay, so that's what these guys look like. But the idea is if you have extra chromosomes, like we saw with our chromosome squash, you probably remember before heading off the break, some of you guys had an extra chromosome, some of you guys had a Down syndrome, some of you guys had a Patau, some had an Edwards. But if you have an extra chromosome or a missing chromosome, that's typically not a good or a favorable position to be in. Some organisms can handle it. We, however, cannot. We have very little tolerance for extra DNA or missing DNA. So it's almost immediately catastrophic for us. So it's very rare for us to see individuals with missing chromosomes or with extra chromosomes. It's not that we become mutants, it's that we just die. There's so much information on the chromosome that if we are missing one, it's just a catastrophic fail when it comes to development and just being able to run our normal physiology. So when you take a look at a chromosome itself, the chromosomes comes as chromatin, right? We kind of talked about this a little bit before, but remember the chromatin is basically this DNA that's wrapped around these little histone complexes, like drum-like complexes, 8 of them. And your DNA is wound around each one of these, and each one of these is referred to as a nucleosome. So that's typically what a non-dividing cell looks like. So the DNA is in this sort of what's called chromatin structure. So it doesn't have a lot of structure to it. It's kind of a little on the loose side. There's, however, different, now that we've studied this a lot, this has actually become a very hot area of research actually in the past. But this basic structure, which is just basic kind of DNA and protein structure, can actually be packed either tighter or looser, and they can give you different textures of DNA. So we have 2 different types of DNA texture types in a non-dividing cell. The first one is what's called heterochromatin. So this is really condensed. And tightly packed chromatin. So if you take this structure up here and you kind of wad it together tighter and you kind of wind it up tighter and tighter and you sort of pack it all together, then it becomes inaccessible. It's not expressed because it becomes inaccessible. And so as a result, these tend to be what's called transcriptionally dark. So if there's a gene on there in that area, it's not going to be expressed. That means it's not going to be able to make its protein. So it'll be shut off. It's an effective way to silence that gene. However, there are areas of euchromatin which are expressed. So this is basically looser packed chromatin. Where genes are accessible. And we actually have zones on the chromosome now that we know of this. So we actually have areas of heterochromatin and euchromatin. For instance, on a chromosome, so here's your centromere, the constriction point associated with the chromosome. So here's your long and here's your short. So in and around the centromere is areas of heterochromatin. Then also at the tip is also hetero. In between is euchromatin. So most of your genes are in this in-between region. So if you look at the topology of a chromosome, like where things are, in and around that centromere is a heterochromatic area of varying degrees. Now, is it permanently heterochromatic? No, because just like you can pack this chromatin down to make it accessible, you can also unpack it to make it more accessible. So you can actually go back and forth and back and forth. We're starting to realize now that this difference between chromatin and what we call chromatin modeling, either condensing the chromatin into an inaccessible package versus loosening it up to an accessible form, is a major way that genes are regulated in whether or not they actually express their particular code. So you can turn a gene off literally by just packing it so tightly that it's inaccessible. Right. So that's the chromatin piece of it. And of course, you have your nucleosome, which is essentially this little 8 histone drum with your 200 nucleotides of DNA wrapped around it. And the reason why it gets wrapped around it is because the histones themselves that make up this little drum are positively charged, and so that negatively charged phosphates in the DNA are attracted to it, and so they kind of attach to those histones. And so this is kind of what it kind of looks like. So here you can see your DNA molecule. And so you would never work with a DNA molecule like this, right? Because the DNA molecule just by itself is super fragile. So if you were to grab onto this, it would basically shred it into a million pieces. So you always want to wind it around your histone core 2 times, and then you kind of hold it together with another histone right there, like a little pin. Kind of stick it right in there, kind of hold it all together. And this one piece right there, that's the nucleosome. That's what's called the nucleosome. That's what it normally looks like. So the other thing that we want to do is we want to basically take a look at the DNA structure, and we want to start moving toward packaging the DNA in such a way that's going to allow us to be able to move it around, right? Because we already seen that part of mitosis is separating the DNA. You have to move the DNA physically into 2 separate piles. So the first thing we need to do is make sure we put the DNA in a structure that can tolerate that movement. Okay, and that's what we're going to start off with nucleosome. And the 30 nanometer fiber. We'll start off with that one next time because we're at about everything else. And none of that, but I feel like my voice is about ready to collapse in a second.